Então, vamos recomeçando. Então, boa tarde a todos. Eu sou a Ana Paula Davel, secretária da FeSBE. Eu só queria dar alguns recadinhos que eu tentei dar no início, né, Léo? Mas por problemas técnicos de criança de 3 anos em casa, não consegui. Agora estou mais tranquila aqui. Então, só para falar que essa atividade de hoje faz parte das atividades pré-congresso da FeSBE, pré-reunião anual da FeSBE, né, a segunda atividade pré-congresso. Na semana que vem nós teremos outra atividade que falará sobre os horizontes da pós-graduação no Brasil, ou seja, né, um tema para bastante discussão. Só lembrando a todos que vai se iniciar às 4 horas da tarde na semana que vem, tá? E a gente vai contar com coordenadores das áreas de ciências biológicas na CAPES, então vai ser uma discussão bem enriquecedora. E mais um informe que eu queria dar para vocês, e o último, é sobre a extensão do prazo para submissão de trabalhos na FeSBE. Então, nós temos mais essa semana de late abstracts, então tem até o dia 15/8, para quem ainda não submeteu seu resumo, submeter, e para quem quiser participar do congresso, as inscrições estão abertas, é só participar. Então, obrigada, Léo, obrigada a todos.
Obrigado você, Ana, novamente, viu? So let's move on to next speaker. My pleasure to invite Professor João Paulo Camporez, who is professor at the School of Medicine from University of São Paulo, campus of Ribeirão Preto. Dr. Camporez has an impressive CV with a list of high-quality papers published. Also, it's important to mention here that he works at Yale University, US, for 7 years or so, under supervision of Professor Gerald Schumann, who also is a very important name on the field. And today, João Paulo is going to talk about the effects of estradiol on non-alcoholic hepatic steatosis. So, João Paulo, thank you very much for coming. And feel free to get your talk started.
Léo, eu acho que eu vou ter que sair do Zoom para permitir a apresentação.
Ok.
Oh, no, I think it's okay now, right? So, can you see?
Yes, you can.
Okay. So I would like to thank Professor Leonardo and the organizing committee for inviting me for this presentation. I feel I'm a little outsider here, so I'll give you some a little spoiler. I'm not going to show you any mitochondrial data today. But I hope I'll show you some interesting data about this study, that it's an ongoing study in our lab. It's about estradiol effects on non-alcoholic steatohepatitis, or NASH. So non-alcoholic steatohepatitis, or NASH, it's an evolution from fatty liver disease. Fat liver, that it's just a simple steatosis, and NASH, its evolution. It presents hepatic steatosis, ballooning, inflammation, and fibrosis, as you can see here. This is the characterized NASH in liver. But why is it important to study NASH? As you can see here, NASH, it's the chronic liver disease that most increased its prevalence since 2002. And today, you can see that NASH is the second most important cause of liver cancer. And consequently, it's the second most important cause of liver transplant. So here you can see the NASH, one of the characterized of NASH, it's hepatic triglyceride content, and you can see that increased hepatic triglyceride, it's associated with insulin resistance in human. So liver tag, it's associated with hepatic insulin resistance and increased hepatic glucose output, which leads to fasting, hyperglycemia, and type 2 diabetes. So liver fat leads to increased diacylglycerol in liver, which leads to activation of protein kinase C epsilon or PKC epsilon. Its activation leads to inhibition of insulin receptor and consequently hepatic insulin resistance. Estradiol also, it's associated with fat liver. We can observe that knockout mice for aromatase, so knockout mice for aromatase don't have estradiol because they cannot synthesize estrogens. So knockout mice for aromatase develops fat liver, as we can see here. Also, we show that estradiol treatment in ovariectomized mice can improve insulin sensitivity in these mice, as you can see by the increase the glucose infusion rate during a hyperinsulinemic euglycemic clamp. This improvement in insulin sensitivity was associated with reduced hepatic tag content and also muscle tag content. So next we asked, are female mice protected against high-fat diet induced insulin resistance? So we studied male mice and female mice fed a regular chow, and we did a hyperinsulinemic clamp, euglycemic clamp, and we see no difference between male and female. But we know that male mice fed a high-fat diet for a few weeks, they develop insulin resistance, as we can see here, by reduction in glucose infusion rate. But female mice, after a few weeks of high-fat diet, they didn't develop insulin resistance. They were protected against high-fat diet induced insulin resistance. This protection was associated by less liver tag content, as you can see. Male mice fed a high-fat diet display less fat liver. Also, female mice fed a high-fat diet display less muscle liver, muscle tag, as you can see here. Next, we asked if estradiol was the cause of the protection in female mice. Then we treated male mice with estradiol and fed them with high-fat diet. And then we observed that male mice treated with estradiol display improvement in insulin sensitivity, as we can see here. This was also associated with reduced tag, hepatic tag content, and reduced the muscle tag content. A few years ago, it was published that ApoE knockout mice fed for 7 weeks of Western diet, they developed NASH. So this is an experimental model of NASH. It's an ApoE knockout mice fed a Western diet for 7 weeks. So we study to better characterize these mice, we study male mice, male ApoE knockout mice, and fed them with a Western diet for 7 weeks. And we saw that they develop obesity with increased body weight and increased body fat. This was associated with increased glucose intolerance, as we can see during a glucose tolerance test. Also associated with increased hepatic tag and increased in plasma hepatic enzymes, ALT and AST. These mice also display increased fibrosis in liver and increased inflammation in liver. So next we asked, what does it happen in female mice? Then we study female mice fed regular chow. These are the control mice, sham. And control mice, sham mice, fed a Western diet. And the overactomized mice fed the Western diet. They were fed with Western diet for 7 weeks as well. The body weight, we cannot see any difference in body weight, the final body weight after 7 weeks of Western diet, but we can see increase in body fat just in the OVX mice, as you can see here. This was associated with glucose intolerance, as we can see in the OVX mice. Also, we can observe that sham mice fed a Western diet didn't develop glucose intolerance. They were protected against the glucose. The Western diet induced the glucose intolerance. These data were associated with increased liver fat in these mice, increased plasma ALT, and increased plasma AST, suggesting liver injury. So we also can see that the liver, the OVX mice fed a Western diet display liver inflammation, as well liver fibrosis. And the sham mice, the control mice, the intact female mice fed a Western diet were protected against Western diet induced inflammation and fibrosis. So we treated the animals, the OVX mice, with estradiol and fed them the Western diet for 7 weeks. So there is a subtle reduction in body weight in the mice treated with estradiol after 7 weeks of Western diet, and a big decrease in total body fat. Estradiol treated mice also display improvement in glucose tolerance, as we can see during glucose tolerance test, and also reduce the hepatic tag content and muscle tag content. So the summary and conclusion so far, because it's an ongoing study, it's that female mice are protected against diet induced the insulin resistance. This protection seems to be estradiol dependent. Estradiol treatment protects male mice against diet induced the insulin resistance. OVX mice display accelerated development of NASH under Western diet. and estradiol treatment protect OVX mice against Western diet-induced NASH. Taken together, these data support the hypothesis that estradiol protects OVX mice from Western diet-induced NASH and glucose intolerance by protecting female mice against hepatic steatosis, inflammation, and fibrosis. So I would like to thank the financial support, FAPESP, CNPq, and CAPES, the collaborators from FMRP-USP, and also the members from my lab here in Ribeirão Preto, our grad students. They are the ones who are working hard on this data. So thanks.
Thank you so much, João. Now we are open for question.
I have one question. Very nice data, João Paulo. Thanks. Did you measure estradiol in these obese mice? And another question that I have is, do you think if you fed these female mice for a longer period, for example, I don't know, more weeks, this estradiol protective effect will be lost? Maybe the effects via ER, via estrogen receptor will be changed. What are you thinking about this scenario?
The first question, we have not measured estradiol in the obese mice. We may expect a little increase because of the increase in adipose tissue, but we are not sure about that. The second question, I believe that eventually the protection is gone, but not. It's not like the male. They develop insulin resistance under high-fat diet condition very fast. Even with 3 days of high-fat diet, male mice develop insulin resistance, but female mice takes a little longer to develop this. But eventually they will develop insulin resistance, but I think it's always a little less than male mice.
Okay, thanks.
João, you have a couple of questions here in the chat. The first one comes from Carlos. Great talk, João. How estradiol would decrease body weight and improve glucose homeostasis? Any clue or any hint about that?
Thanks, Carlos, for the question. Yes, a few groups see a subtle effects on feeding, but I never saw this. What we see, and I think most of the people who study estradiol and metabolism, it's the estradiol increases energy expenditure. So my first paper showed that increase OVX mice treated with estradiol increase the energy expenditure. So they increase oxygen consumption, basically.
Now you have another question. It comes from Tanis. Are the phenotypes for somehow associated with this estrogen receptor activation?
There are some evidence that show that estrogen receptor alpha activation is associated with these estrogen effects on metabolism. So we have the estrogen receptor alpha flux mice, and we are going to study the liver specific knockout mice. So yes, it's true, it's alpha isoform of the receptor. By the way, there are two estrogen receptor isoform, alpha and beta, and it's through the alpha isoform, its effects.
Okay. João, I have a small question for you now. Have you tried to measure metabolic profile of the mice, male mice under estradiol treatment?
Yes.
Like, for example, oxygen consumption?
Yes, we did. And we saw increasing in oxygen consumption. I mean, whole body oxygen consumption.
Okay.
It's the CLAMS study. There is no effect on physical activity and no effects on feeding, but there is improvement, increasing actually in oxygen consumption energy.
Okay.
So maybe that's why they decrease liver, muscle fat, and consequently improving insulin resistance.
Thank you. Anybody else? Okay. Thank you so much, João Paulo, for taking your time, participate of this meeting with us, and congratulations for a wonderful talk. Thanks a lot.
Okay, thank you.
So let's move on to last talk of today. My pleasure to invite Dimitrius Santiago, who is a PhD student in my lab. Dimitrius has an academic background in biochemistry with expertise in molecular biology. He has a set of new findings in which he is proposing that the transcription factor NR2F6 is an important regulator of mitochondrial metabolism. So Jimmy, thanks a lot for participating, and feel free to get your talk started.
Okay, so thanks, Leo. Can you all hear me?
Yeah.
Okay, then. Let me start sharing. Okay, then. So first, I would like to thank you all for sparing a little time for hearing a little bit more about the regulation of mitochondrial metabolism. We really appreciate this time here, and I'm really glad for presenting our work today. I hope the presentation is worth it. So enough small talk. So I'm a PhD student, as Leo stated. In our lab is located at the Obesity and Comorbidities Research Center here at the Institute of Biology in UNICAMP. And therefore, our general goal is to understand the pathways that lead or worsen the metabolic syndrome. And more specifically, Leonardo groups is interested in the regulation, actually the transcriptional regulation of mitochondrial genes, and primarily in insulin resistance. And one of this family of transcription factor that is classically pointed as a major regulator of mitochondrial function is the nuclear receptor family of transcription factors. So as you can see here in some of our publications, we get a focus on the PPAR beta and delta and its relation with the PGC1 alpha, which is a classical regulator, master regulator of mitochondrial oxidative metabolism. Well, one of these nuclear receptor that caught our attention, and it's a nice coincidence that Professor João Paulo spoke a little bit about NASH disease, because the nuclear receptor, an subject of our research, has been recently pointed as a marker and a regulator of accumulation of lipids in the liver. Last year or 2 years ago, it was shown that it is upregulated by high-fat diet in the liver of mice, and that its knockdown can actually prevent overall accumulation and development of NASH disease. So using also prospecting the NRF6 regulated genes, we were with a public available data set, we were able to see that, we were able to see that NRF6 sistrum in HIP-G2 cells, which are human hepatocarcinoma cells, is enriched with genes related to the metabolism of lipids, insulin resistance, and even genes located to the mitochondria. And so we wondered whether NRF6 could also be related to the lipid metabolism in the skeletal muscle. And the first step was to check the NRF6 expression and protein content in the gastrocnemius muscle of mice fed with high-fat diet for 12 weeks. And strikingly, the levels of NRF6 were reduced in this in vivo by lipid overload, which could also be reproduced or mimic in vitro experiments with C2, C12 myotubes treated with palmitate or a blend of palmitate and oleate for 24 hours. So we asked whether this reduction in NRF6 in skeletal muscle was an adaptive response or actually contributing to the development or worsening of muscle insulin resistance, which is our main interest. And therefore, we isolated this variable and performed a fatty acid oxidation assay with knockdown myocytes for NRF6. And this oxygen consumption assay demonstrated that myocytes depleted of NRF6 presented higher rates of oxidation of fatty acids in oxygen consumption experiment. And so can these cells be protected against insulin resistance? So because, well, they can oxidize palmitate more efficiently. So can they be protected against palmitate-induced insulin resistance? So we used stable cell lines knockdown for NRF6 and verified that even with palmitate treatment, these cells maintained AKT phosphorylation induced by insulin. Moreover, these cells were less prone to palmitate-induced cell death and were protected from superoxide formation. So these strong phenotypes led us to ask which mechanisms or molecular pathways could be involved, since NRF6 was not implied in this type of regulation in skeletal muscle. Actually, it's the first time we've seen NRF6, this nuclear receptor function in the muscle metabolism. And what we've seen was that several genes responsible for the oxidation of fatty acid, such as CPT1, FABP3, were being upregulated by NRF6 knockdown in C2, C12 myocytes and mouse primary myotubes. And one of these genes is the uncoupling protein 3, which is, I can say it's our gene of interest, given its function in the reduction of a reactive oxygen species formation in the mitochondria and its role in lipid oxidation. Since we've seen both these phenotypes before, as I previously showed, we verified if NRF6 could directly regulate UCP3. So using, again, public available SHIPSEC data and bioinformatic tools, we found an NR2F6 response element on the UCP3 promoter at the same site that myogenin and MyoD proteins bind to the UCP3 promoter, meaning that this could be direct regulation of UCP3 expression by NR2F6. And trying to gather more evidence for this, we performed reporter gene assay with luciferase expression under the control of the UCP3 promoter. And what we found was that actually the NR2F6 knockdown could activate the UCP3 promoter, further indicating that NR2F6 directly knockdown directly de-repress UCP3 expression, which could explain our previous results. However, since it's again a novel pathway for UCP3 regulation and lipid metabolism in the muscle, we performed a RNA-seq experiment to further understand the molecular signature of these NR2F6 depleted myocytes. And we've seen that several genes of the insulin signaling pathway were being modulated by the knockdown. As you can see on your right, the AKT basal AKT phosphorylation levels were higher, and also AKT activity as seen by the AKT substrate GSK3B. Additionally, these cells also presented higher phosphorylation of the AMP kinase, and several genes downstream from this kinase were also being upregulated in this case, which could explain together, these results could explain how these cells depleted from NR2F6 presented a resistance to the insulin resistance induced by palmitate. But the most striking result was that the NR2F6 knockdown actually was concertedly modulating quite a specific set of genes. One of these genes being downregulated were mostly members of the cell cycle ontology and DNA transcription silencing genes. And on the other hand, processes related to the metabolic processes in muscle differentiation were being upregulated. Moreover, if we note on the heat map on your right, several markers for muscle differentiation were also being upregulated. So, so far, our results, our results can indicate two types of regulation of the muscle oxidative metabolism by NRF6. First, the first of them is a direct derepression of NRF6 targets by the knockdown, but also since several of these oxidative genes are also increased during the myogenic differentiation, NRF6 could also be acting indirectly by increasing the transcription of several muscle specific genes such as myogenins, and therefore increasing the levels of the UCP3, for example, which could be responsible for the observed phenotypes. So I kept this presentation short, and all of your questions are very welcomed. I would like to thank again for the financial support of the agencies, and if anyone has any questions, you can contact us later by at these two email addresses. So thank you very much.
Thank you, Jimmy. Questions?
I have a question.
Please, Tanis.
So yeah, great work, Jimmy, and really nice data. And my question is whether you used myoblasts or myotubes for the analysis, the RNA-seq analysis, the phenotyping and everything.
So thanks, Tanis. So this is that myocyte, they are not actually fully differentiated myotubes, which could also help explain the strong impact of on the cell differentiation genes. So they are myocytes at the third day of differentiation.
Okay, because the RNA-seq data that you show seems that you have a downregulation of this cell cycle, right? Cell cycle targets. And normally when this happens in C2C12 cells, it boosts differentiation. So it's kind of associated. When you block the cell cycle, you get more differentiation for myotubes formation. So one of the questions that can come up here is whether the effects that you see both in mitochondria and the targets and everything could be associated with increased differentiation by the decrease in the cell cycle genes.
So yeah, that's a really great question. Actually, I believe so. At this point, I believe that NRF6 is actually acting as a fulcrum, balancing a cell cycle and the myogenesis. As you said, they are opposing processes. And I've not brought us here, but the myoblasts depleted of NRF6 are actually proliferating at the lower rate than control cells. And I've seen that some genes related to the cell cycle, such as P21 and RB1, which are actually related to the cell cycle arrest, are up-regulated. And more striking, work being developed by Ninon, which is a great contributor of this work, with inertia 6 overexpression cells, have shown that these cells can proliferate at higher rate, but we are verifying now whether they have also a block or a reduction in cell cycle arrest genes.
Okay, cool. It'll be nice to check this in the mice, right? In vivo. That'll be the greatest.
Yeah, we are trying to import these mice now. They are actually would be shipping for us next month, I hope. And we are also performing some in vivo experiment with Rafa
okay.
Dr. Isis.
Yeah, nice. Okay, cool. Thank you.
Thank you so much.
Anybody else? No question. Okay. Thank you, Dimitrius, for our very clear presentation and very warm findings. Thanks a lot.
Thanks you.
So guys, that's all for today, and I hope you enjoy. Once again, thanks a lot for attending, including our speakers. And I really hope to see you next year. Hernandes, feel free if you want to say some words. Thanks a lot.
Muito obrigado a todos. Esse foi o nosso segundo encontro do mês de agosto. Eu gostaria de convidá-los para participar dos próximos encontros que nós teremos. Na próxima quinta-feira o tema será Horizontes da pós-graduação nas ciências biomédicas, com os coordenadores das 3 áreas da CAPES. Nós estamos convidando coordenadores de pós-graduação para termos um debate bastante produtivo, no sentido de entendermos para que lado estamos caminhando com a pós-graduação de um lado e o sistema de avaliação de outro, certamente. E no dia 26 nós teremos um simpósio sobre análise de células únicas, ou single cell RNA-seq, que também está prometendo ser bastante interessante, uma tecnologia nova, bastante útil em vários aspectos. E, obviamente, esses 4 encontros, eles são um aquecimento para o Congresso da FeSBE, que vai acontecer de 8 a 11 de setembro, que eu espero contar com a presença de todos vocês e com a contribuição no sentido de divulgar para que nós tenhamos bastante gente se inscrevendo nos próximos dias. Então, eu agradeço a todos, agradeço ao Léo por ter proposto essa atividade, que foi extremamente importante e de alto nível de discussão. Muito obrigado a todos, até breve.
Obrigadão, Hernandes. Tânis, obrigado. Ângela, obrigado.
Valeu,
João Paulo. André, Dimitrius, obrigadão a todos vocês.
Tchau, pessoal.
Os participantes também. Valeu, Tânis.
Valeu.
Valeu, João. Obrigado.